hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat, lilipad. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat pagtulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparana sa bayang Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ang aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap-iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap-iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Magandang araw at magandang agham po sa ating lahat. Welcome to the 18th installment of our Virology and Vaccine Institute of the Philippines, or the VIP webinar series. I am Joseph Sonyo from the Environment and Biotechnology Division of the DOST ITDI, and I will be your host for today. This webinar series is part of the Virology and Vaccine Institute of the Philippines, or the DIP Information and Education Campaign on virology and other related topics. These learning sessions made possible by the collaborative effort of the DOST Central Office, DOST ITDI, DOST PCHRD, DOST PICARD, and DOST Balik-Scientist program. I am sure that everyone here is excited to learn. Am I right? So let's check first the number of participants here in the Zoom meeting, as well as our audiences from Facebook and YouTube Lives. So we have almost 100 in our Zoom meeting room and more in our Facebook and YouTube Lives. So before anything else, I would like to thank everyone for coming in and joining in. And just a reminder for our Zoom participants to kindly rename your Zoom account to this format: affiliation _ your name. Like, for example, me, DOST ITDI _ Joseph Sonyo. So we could easily identify and properly address your question later on. Lastly, don't forget to mute your microphone to avoid interruption during presentation. So, before we formally start today's webinar, let us share first a couple of video presentation to give our participant an introduction to the VIP program as well as the Balik-Scientist program.
Recognizing the critical role of science and technology in economic development and progress, the Balik-Scientist Act, or Republic Act 11035, was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago, but we need some legal support so that we can implement it in a better way. The Balik-Scientists will have easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik-Scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom to our own institutions, our own researchers.
My role and responsibility of a Balik-Scientist is to be not just a teacher or a facilitator, but also a pusher of innovations within the program.
Balik-Scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik-Scientist program, is tasked to facilitate the placement of the Balik-Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik-Scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik-Scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik-Scientist program. Okay, being one, I really truly felt that Balik-Scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik-Scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik-Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people.
All right, so that was our video about the VIP program and also the briefer for the DOST Balik-Scientist program. Thank you so much, DOST ITDI, for preparing the clips. And now, before we start, let's first have a recap of the previous webinar last Monday, February 14, hosted or delivered by Dr. Epidio Cesar Nadala, one of our Balik-Scientists from the United Kingdom. He gave a topic on virus and host interaction. He lectured about the mechanism involved in host infection, as well as abilities of the viruses to evade host defenses. He also gave an insight on how the virus interact with our system molecularly and immunologically. Now, for today's webinar, we shall be learning a lot about molecular virology, genome sequencing, and bioinformatics, which will be discussed by our resource speaker, Dr. Christina Leyson, a Balik-Scientist from the United States. So to formally introduce our distinguished Balik-Scientist and resources speaker this morning, she obtained her bachelor's and master's degree in molecular biology and biotechnology at the University of the Philippines, Diliman. Thereafter, she pursued her doctorate in infectious diseases at the University of Georgia. During her master's study and as a university research associate, she worked on a diagnostic of a Baka-Banchitap virus at the National Institute of Molecular Biology and Biotechnology at the UP Diliman. Dr. Leyson was a guest lecturer and a teaching assistant at the University of Georgia for the topics on human anatomy and physiology, avian viral diseases, veterinary bacteriology, molecular virology, and introduction to virology. At present, Dr. Leyson is a postdoctoral researcher on exotic and emerging avian viral diseases group at the Southeast Poultry Research Laboratory of the U.S. National Poultry Research Center, an agricultural research service in the U.S. Department of Agriculture. Armed with her experience in DNA, RNA, and protein laboratory techniques, including quantitative PCR, cloning, and protein expression, knowledge on sequence analysis, phylogenetics, and homology modeling, she was able to publish dozens of research papers in international peer-reviewed journals. She also serves as a research speaker in several conferences in various countries like the United States, Japan, and of course, the Philippines. So now, let's give a warm welcome and a big virtual round of applause to Dr. Christina Laura M. Leyson. Dr. Leyson, the virtual floor is yours.
Hi, good morning po sa inyong lahat. Gabi po dito sa US. Thank you so much for that very kind introduction. Can you hear me? Okay. Yes. Okay, great. Okay, so again, thank you so much for joining me. There's 141 participants on Zoom, so I'm very happy about that. Hopefully, I will make great use of your time and we'll learn together. So let me just start sharing my slides. Okay, and then if there's any questions that you have, please let me just put it in the chat. I'll do my best to monitor the chat. Okay, great. Can you see my slides okay? Okay. So no complaints means silence means yes. So I'll just get started. Okay, great. So for today, I will give like I titled the presentation to Intro to Molecular Virology. So hopefully, you'll learn something new for today. Okay, so I like to start talking a little bit about myself, parang a little plug for virology. So it all started with a bacteriophage. I was in a class at UP Diliman for my undergrad. We learned about this bacteriophage, and I just thought it was the most fascinating thing that this small piece of DNA can override the whole cell and make more of itself. And so from there, I did my thesis on a Baka-Banchitap virus, which is, hopefully you can see my mouse. Let me make it, oops. Hopefully you can see out, which is a baka, which looks like a banana, which harvested for its fiber, but I worked on the virus that makes it smaller. So yeah, and then I also worked with rabies, and for the past 8 years or so, I've been working with avian viruses, which I talked about in other series. But for today, I'll be talking about a lot of my molecular background and also something that I've really been working on for the past year, which is virus evolution. So hopefully you'll find some use there. So before we start, I like to get to know you as well. So I already said stuff about me. So I'm gonna go ahead and stop this presentation and go to another presentation called Mentimeter, because I would like to learn more about you. Whoop. Okay. So even if we're so used to Zoom, apologize. Okay, here we go. All right. Okay, so if you go, I would like to learn more about you. So if you go to Mentimeter, so menti.com, and then put the code that's on the top of the slide. And if you go ahead and answer the questions. So number 1 is, am I an early bird? Agree or disagree? I like milk tea. Agree or disagree? And SARS-CoV-2, agree or disagree? So I'll give you a couple of minutes, or a couple of seconds, I should say, to answer the question. Okay. Oh, most people are early birds. Maybe. That's actually an I'm a little bit of an early bird. I do sleep kind of early, but I know that is not most people. All right. More people are coming in. Give you a few more seconds. So we're trying this platform out, because I like to also ask you for feedback and see if we can, like in the middle of the lecture, to also get feedback from the audience, because this is Zoom land and I can't see you. So I'd like to hear some more. So maybe like 10 more seconds, maybe more people can come in and answer the question. Maybe when you hit 40 participants, so maybe 3 more. All right. Okay, so I think we have middle. So I'm an early bird. Some people are not, which is expected actually, in the middle. I like milk tea, also in the middle. I'm surprised by that. I'm assuming that most people like milk tea. But yes, I'm wrong. And then the last one is just actually a little bit of a diagnostic test. So SARS-CoV-2 has a DNA genome. And yeah, most of you are right. So actually SARS-CoV-2 has an RNA genome. Yeah, we'll talk more about SARS-CoV-2, because I think that's something that we are all interested in. Okay, great. Thank you so much, guys, for indulging my little experiments. So let's go back to the presentation. Hopefully I will manage this. Okay. All right. Okay, so thank you for that, guys. All right, so let's go to the outline of the lecture. So the first 2 parts is actually a review, which I took for a lot from and added a few things from my previous lecture about animal viruses. If you haven't seen that, you can totally check it out. And then we'll review what are viruses. So basically, I think, and also how do we classify viruses and virus genomes, because these are really fundamental information to be able to understand all the other stuff, especially evolution and phylogenetics. And then lastly, we'll use everything that we've learned about sequencing and evolution and phylogenetics, and looking at how do we understand SARS-CoV-2 lineages, because I think it's something it's very useful to know, and it's very relevant to all of us because we are in a pandemic caused by SARS-CoV-2. Okay, so first off, what are viruses? So in Latin, virus means poison, and viruses are obligate intercellular parasites, which have genetic material, which is RNA or DNA. So SARS-CoV-2 has RNA, and it's surrounded by and sometimes by a membrane. And then one important thing actually to note is that not all viruses cause disease. So it may or may not cause disease. And many, I will see later, many infections are actually inapparent. So viruses are everywhere. right? And so it's kind of like this idea that bacteria are also everywhere. So, and we live and thrive in a cloud of viruses, and we, for the most part, do fine. So most infections have no consequence, and if we do get infected, many infections are apparent. And here's just an example of just a fun fact that the most abundant virus in wastewater, so people that are the stuff that are in feces and urine, is actually not an animal virus. It's called a pepper mild mottle virus. And it infects peppers, and it's probably really super stable, and that's why it's one of the most abundant viruses in wastewater. So yeah, fun fact. And it's everywhere, and it doesn't affect us, even though it's in our waste, bodily waste. And so viruses are obligate into cellular parasites, and there's many lifestyle, right? There's many different kinds of viruses. There's many different kinds of pathogenesis. There's also there's just so many different lifestyles that viruses have. And so for every solution, it must reveal something about the host as well as the virus. So just food for thought. And by the way, these a lot of these slides are from Vincent Raccaniello's virology lectures. I encourage you to check them out. They're all on YouTube and in his blog, I believe. All right. So that is the part about classifying viruses. So what are viruses? Yes, TWIV. TWIV is fabulous. And that's from someone from UBLE. Didn't catch. Okay, great. So how do we classify viruses and virus genomes? So first off, what is a genome? Right, so we're starting with the basics. So a genome is the collection of all genetic information of an organism. And then in practical terms, this is represented by a string of letters that encode the genetic information. So DNA and RNA can be composed. You can think of it as a really long word composed of only 4 letters, each letter representing one base or one type of base in the genome. And it can be an A for adenine, G for guanine, C for cytosine, and T or U, T for DNA, and a uracil for RNA. And those are the 4 bases. And so here's just this is a real example on your left of an actual sequence. It's just basically a really long word, and that represents the genetic material of the virus. Okay, so the discovery of DNA as the genetic material was actually discovered from viruses, and these are called bacteriophages, or viruses that cause, or I would say that infect bacteria. So bacteriophages. And so this is a really famous experiment called the Hershey-Chase experiment, where remember, so viruses can be composed of nucleic acid and protein. So in this experiment, they labeled the protein, which is the bacteriophages, in red on the left. And then on the other set of experiments, they labeled the DNA, which is green on your right. And so when they use these 2 types of viruses, these 2 labeled viruses, infect bacteria, and looked at what nucleic acid made it into the bacteria. So what was the part of the virus that made it inside the cell? And it was, as it turns out, the DNA, which is in green on your right. So that's how DNA as genetic material was discovered. And so there's also in molecular biology, there's also this, in quotes, central dogma of biology, which is a very classical view of how DNA or the genome is replicated. And so in this central dogma describes the process by which instructions in the DNA are converted into a functional product. And this is in terms of called an mRNA or messenger RNA, and that eventually gets translated into protein. And in this central dogma, there's actually 3 major events called the DNA replication. So DNA making more of itself, transcription making mRNA from DNA, and translation making protein from mRNA. But as we all know, in biology, there's always exceptions. And what's better microbe to have an exception, but in viruses. So for viruses, it has been shown that viruses can also have a different kind of genetic material, and this is called RNA. And this experiment from Frankel and Conrad showed that genetic materials. They do a very similar experiment with Hershey and Chase experiment, where they disassemble the particles, label the protein part of the virus, and then label the RNA part of the virus. And then from there, they infect the tobacco. So this virus that they used is called the TMV, or tobacco mosaic virus, and looked at which part would cause the which part of the which RNA from the two different kinds of virus will cause like this particular lesions on the tobacco mosaic virus. And then from this, they show that it's the RNA part that if they label the RNA from a particular strain of TMV, the lesion that come out is always correlated with RNA. And they're showing that the RNA is the genetic material, can be used as genetic material, most especially for viruses. Okay, so because like viruses have are really diverse, and there's many different kinds of viruses, there's actually a committee called the ICTV, or the International Committee on the Taxonomy of Viruses, that actually is a group of scientists and researchers that are tasked with developing and refining and maintaining the universal virus taxonomy. And this was formed by the IOMS, or the International Union of Microbiological Societies. And so every year they publish the taxonomy, the virus taxonomy, and you can go to their website, which is talk@ictvonline.org. You can see what the taxonomy of your favorite viruses. And then there is a process for which the taxonomy of the viruses can get ratified, and there's an actual process. So there's a committee. There are different committees and subcommittees for different viruses, because there are many different. If there is any living organism, there is a virus for it. So there are different groups for different kinds of viruses. And every year they decide on whether or not the taxonomy reflects our current understanding of the viruses. And so even though that there's a lot of different kinds of viruses, because viruses infect pretty much every single type of living organism, and there are seemingly infinite complexity of infections, there's actually a finite number of viral. There's only seven viral genomes, 7 different types of viral genomes. And this was initially proposed by David Baltimore, and it's famously called, it's also known as the Baltimore Virus Classification Scheme. And so in this classification scheme, there are 7 different types of different kinds of genomes, and they're all listed here. And it really depends on what kind of nucleic acid is found in the virus particle. And so just to make your life easier, just know this, like all viral genomes, all without exception, at least as we know it now, all viral genomes must make messenger RNA or mRNA. So remember from the central dogma, mRNA is one of the functional products of the genome. Ready to be read by ribosomes. So mRNA is read by ribosomes to make proteins, and all viruses need to go through an mRNA to make proteins. And so, at least as far as we know, right? Okay, so in the Baltimore scheme, all of the 7 different types of viral genomes all converge into mRNA, which is in the middle. In the middle, and that's because mRNA is used to build proteins. And so in this Baltimore scheme, this is important to know, like that you can also appreciate that there are different pathways to go through mRNA. So some viruses go from positive RNA to negative RNA, and then again to the mRNA. There are some double-stranded RNA viruses that go straight to mRNA, and so on and so forth. And this, these kinds of like looking at what type of viral genome a virus has different implications. One, because it indicates what kind of lifestyle the viral genome has, and on the more practical side, viral genome, like the viral genome, can also is an important consideration when you're making antivirals. So, for example, if you have and here's an example where if you have a drug that targets RNA-dependent RNA polymerase, then it's less likely to affect the humans, for example, because our genome is DNA. So it's a DNA dependent, is DNA. So we're less likely to be affected by that. So that's just an example of how the viral genomes can be important. Okay, does anybody have any questions so far? Just put it in the chat. I will try to monitor it as best I can. So some important definitions. So again, the mRNA is a ribosome ready, and it's always called the plus strand. So this is just a nomenclature. So the mRNA is the plus strand, and so the DNA equivalent in polarity is also plus strand. And then the complement of the plus strand is the negative strand. So RNA and DNA complements of the plus strands are called negative strands. And one thing to remember, even though the polarity of the mRNA and an RNA is the same, not all positive sense RNA is mRNA. So there are specific structural differences between a positive sense RNA and plain mRNA, or an mRNA that's ready to be read by a ribosome. So just some important definitions. And so if you want to learn more about the different kinds of viruses, I would really encourage you to go look at this website called viralzone.expacy.org. It's a great resource, and it's really up to date. These folks really do a good job of being responsive and really putting this website and all the information in there up to date with current knowledge. So if you ever have any questions about what type of viruses, what kind of genomes that they have, etc., go to viralzone.org, expacy.org. Okay, great. So, and that's a review for the basic virology part. So we're gonna go back to Mentimeter and look at our understanding of mRNA. So let's go back to Mentimeter. Okay. If I'm able to. Maybe. Hopefully. Okay. Let's see. I'm just trying to make it advance. Okay. Present. There we go. Okay. All right. So the question is, why is mRNA placed at the center of the Baltimore scheme? So the options are, so you can go and vote using mentime.com. The first option is because all virus particles contain mRNA. The second is there is no specific reason. Third option is because all viral genomes are mRNAs. The third is, so if you're if you can place your mic on mute, I would really appreciate. Okay, great. And then the most popular one so far, because mRNA must be made from all viral genomes. And then the last is because Baltimore studied mRNA. So let's wait for like 40 people. Now let's be more ambitious. 50 people to answer. And see, hopefully more people. 37 people. Maybe more people are inclined to answer. 10 more seconds, and then we'll answer together. Dun, dun, dun. There should be some music. I should have prepared some music. Okay, so so far our most popular answer is because mRNA must be made from all viral genomes. Our second most popular is because all virus particles contain mRNA. Okay, so actually the correct answer is the most popular because mRNA must be made from all viral genomes. So remember, mRNA is the building blocks, or is literally the message, or the information in mRNA is used to make proteins, and to make virus particles, to make more viruses, we need proteins, and so that's why the fourth option is the correct one. And then remember, like for the first option, the reason why it's not correct is that, remember, there are 7 different kinds of virus genomes, right? So it can be DNA, it can be RNA, it can be single-stranded RNA in the positive sense, or in the negative sense, it can be double-stranded RNA. So it's not necessarily that virus particles contain mRNA. In fact, actually, virus particles not contain mRNA at all. And then the third most popular one, because all viral genomes are mRNAs, is for the same reason. There are, remember, the Baltimore scheme has 7 different kinds of viral genomes, and they all converge in mRNA. Okay, hopefully that is clear. So if you have any questions, again, you can put it in the chat. Let's go back to the PowerPoint presentation. Okay, so for the next part of this first part of the talk is we're going to talk about sequencing technology. So how do we actually, what are the technologies used to determine the virus genomes? And so there are many different kinds of sequencing. In the recent years, if you notice, actually starting 2005 up to now, there's like a boom of different kinds of different sequencing technologies. It used to be just Sanger sequencing. We'll talk more about that later. But really starting in the early 2000s, and really this is correlated with the Human Genome Project, and also the development of companies that commercialize different sequencing technologies. So yeah, we'll talk more about that. So there's actually, I'm going to talk about two main kinds of sequencing, and this first one is the more traditional one, but it's still used until now. I mean, this is oldie but a goodie. I say it's still used, but of course it also has limitations, right? So this is called it's also a determination method. And in this method, the like the fragments are randomly amplified using what's called a reagent that's called DDNTPs, or dideoxynucleotides. We'll talk more about that. And these are very similar to DNTPs, which are the building blocks of DNA, but they terminate the chain. And so what you have is like randomly generated fragments. Hopefully you have fragments that end at every single position on the DNA. And then you do electrophoresis and you detect the fragments either using the radioactively labeled DNTPs, which is not used anymore because it's radioactive and it's not that safe because it's radioactive. But what most people use right now is fluorescence, or a very very sensitive machine that will detect a pH change, and that's using the ion torrent machine. And this is just a picture of Frederick Sanger, who invented this technique, and along with Walter Gilbert won the Nobel Prize for the chain termination sequencing. And like I said, this technique is still being used. And so the key chemistry that's important is the dideoxy ribonucleotide. So what that means is that there are no, hopefully this will not bring back like bad memories of organic chemistry, but usually in normal DNA there is at the 3' position, but the dideoxynucleotide does not have it. If that doesn't make sense to you, essentially the dideoxynucleotide prevents further replication of the DNA strand, and that's why you get strands like this. So if you do this enough, you will get different fragments that stop, that are, each fragment stops at each position on the DNA. Right, and if you run this on the gel, so that creates different lengths, right? And if you run it on what's called a gel or a capillary gel, it will separate these fragments by size, and then the DNTPs are actually labeled, and it's detected by a very sensitive laser. And what you see at the end is something like this, with the peaks corresponding to the DNTPs at the end of each fragment. And then from there, you can read the sequence, essentially like a really long letter, like a very long word just composed of 4 bases. All right. Okay. So that's Sanger sequencing. Okay, so and then the next type of sequencing that I would like to talk to you about is what's called high throughput sequencing, and it's essentially what you think it is. So it's basically just doing sequencing, but a lot of it, like a lot more of it. And so high throughput sequencing techniques allow for highly scalable sequencing. So if you compare Sanger sequencing, when you're just doing one reaction at a time, in high throughput sequencing, the reads can be millions or billions at the same time. And that's why you can generate a lot more data in a much shorter amount of time, and also relatively cheaper. And so these techniques have been developed in the mid-1990s. And then it was commercialized in the early 2000s. And then after then, there are different technologies that were developed. And especially over the past 15 years, the costs were getting lower. And so because of the economy of scale, right? So instead of doing one reaction at a time, you do millions and billions of sequencing reactions at a time. And so for high throughput sequencing, and this is true, like this cartoon, the machines used to take up a lot of space, but it is getting smaller and smaller. We'll see later different of that. So there are two main types of high throughput sequencing: the short read and the long read high throughput sequencing. So yeah, and then before we go into that, here's just like a graph from the NIH, or the National Institutes of Health. They publish this graph and update it every single year. And they're showing here how in starting around 2005, 2006, the costs of sequencing have gone down significantly. And this and right now, actually, we're benefiting from that, especially in this pandemic. I think a lot more people are aware of whole genome sequencing and virus genome sequencing. It has actually allowed for more access to sequencing technologies. Okay, so we'll talk about first short read sequencing. And so there are other technologies that are available, but right now one of the most common is the Illumina platform. So Illumina is a company, and it's one of the most widely used short read sequencing technologies. And their technology is sequencing by synthesis, and it's really not very different from Sanger sequencing. There's just, again, a lot of it. So again, the nucleotides, or the DNTPs, are fluorescently labeled, and every time you do sequencing, or you add a base to the DNA strand, there is a fluorescence that is released. And so what you see, and this is like what an actual experiment would look like. You see different spots with different colors representing the different bases. And so the Illumina like the flow cell where all the sequencing happens relies heavily on very sensitive optics, and it's really small. It's really small, maybe the size of your fingernail, or maybe a thumbnail. And to do sequencing of the sample DNAs, are fragmented. Hence the term short read sequencing. So the DNA is fragmented into fragments about 100 to 200 bases, or maybe more, depending on what specific sequencing reactions you're using. And then these fragments are labeled so that they're barcoded. That's unique for each sample. And this collection of fragments called a library. So a little bit lingo. And then again, because you fragment the DNA, it requires, once you get all the sequencing data at the end, this kind of read will require as assembly, and that assembly happens in the computer. So you get the data, and then assemble it in your computer. And here's just a diagram of how it happens. So again, if you have a sample, the genomic DNA in this case, you fragment. There are different methods for fragmentation. There are enzymatic and physical methods. But the point is, you make different fragments from your genomic DNA, and then you add adapters, which are basically barcodes that is unique for every sample. And then in the sequencing reaction, there is what's called the flow cell, and these flow cells have primers that are complementary to the adapters that you added to your fragments. And what happens is you have what's called bridge, like clusters, which copies your different fragments and makes clusters. So the dots represent the copies of the same fragment. So each dot represents a copies of the same fragment. And then what you do is you denature it, make it single-stranded, and do synthesis. So DNA, basically PCR, on the flow cell using the fluorescent DNTPs. And then from there, every time you extend your strand, there's a release of light from the fluorescently labeled DNTPs, and you read that with super sensitive optics. And in fact, it's actually when you set up your machine, a technician will evaluate your area, your bench area, you're allowed to move it. So you have to because any vibrations or any like jostling will really mess up the optics that is in the flow cell. Okay, so that's short read sequencing. So what about long read sequencing? So a lot of people also call this the third generation sequencing, and it's exactly what it sounds like. So instead of having 200 or 100 to 200 base pairs, and a lot of it for long read sequencing, you can actually have reads or a reaction basically that's can be kilobases or megabases long. And there are two main technologies that are used. One is called the Oxford Nanopore, and then the other one is the Pacific Biosciences or PacBio. And in both cases, it's long read sequencing as of now, which can always change. As of now, is less accurate than short read sequencing, but the advantage is that the sequence assembly is more straightforward. So here's an example on your top right. So if you can imagine, if you only have 4 fragments to figure out how it's arranged, it's so much easier than having like dozens or millions of fragments to assemble together like a jigsaw puzzle. So if you have less pieces, then it's a lot more easier, a lot less computationally intensive to assemble long read sequencing. Also, long read sequencing data also has the advantage of sequencing repetitive regions and also some regions that are really hard to sequence using short read. And it just happens sometimes you get a gap that's that for whatever reason the short read sequencing technology just can't solve. So you can use long read sequencing for that to resolve that area. And so again, like one company that does the long read sequencing is called PacBio, and their methods are pretty similar. So instead of clusters that you have with Illumina sequencing, you actually have pores, and that pore is so small that only one DNA fragment can fit in that. And the detection of the sequencing reaction is also using DNTPs that are fluorescently labeled. And so PacBio also relies on super sensitive optics. So very similar, you'll also get spots where the DNA, the single molecule DNA is, and then you detect the fluorescence. The DNA is using the fluorescently labeled DNTPs. And this, for whatever reason, PacBio machines are pretty big. They're like a fridge. Yeah, I don't know. I don't know why. Okay. All right. And so the other long read sequencing platform is called Nanopore. And at the heart of the technology is actually a protein called the motor protein. So instead of having a hole, so PacBio has a hole, nanopore sequencing relies actually on something that's super similar to what we have in our own cells. So there's a membrane and also the motor protein, and then which takes in your DNA. And as your DNA is being threaded through the nanopore, there is each base has characteristic current change, and so that's read by super sensitive electrodes at either ends of the nanopore, hence the name nanopore. And you get something like this, a data something like this. So each base, for example, at C, it always corresponds to a current change around, I'm going to say that's 90. And then a G is lower, and so on and so forth. So this relies on electric current to read the DNA. All right. Okay, so one thing that's actually pretty cool, I think, about Nanopore, if you noticed earlier how big the PacBio machines are, they're as big as a fridge. So like Nanopore technology is highly scalable. To many degrees, the Illumina center sequencing and PacBio, they can also be scalable. I think the nanopore technology is a little bit more flexible in the sense that you can have a Flongle, which only has one flow cell, or you can have a Prometheon, which has up to 48 cells. And just as a reference, a flow cell is about the size of like a really big USB drive. I'll actually have a picture of it later. Okay, so nanopore technology was used because it's super portable. It was used for sequencing at the International Space Station. So Kate Rubins was the first person to ever sequence in space. And she's a biologist in training, a virologist, in fact, by training, but she's an astronaut now. I guess she's still a biologist because she runs experiments. So Kate Rubins performed the sequencing of virus and bacterial the min ion, and the min ion part. So this is just her computer, the top part, and the min ion part is here, the machine with the barcodes. It's pretty portable. Also relatively cheap because it is smaller. The flow cells are expensive though. That's how they catch you. All right. Okay, and then last but not the least, oh, by the way, Kate Rubins is on Twitter as AstroKate if you ever want to follow her on Twitter. So what makes for good sequencing data? I think this is also important to know. There are two main categories that makes for good sequencing data. So one is depth. So what is depth? Depth is the average number of times a base of a genome is sequenced, whereas coverage is how much genome is covered by sequencing data. And so you can think of it as two dimensionals, right? So the depth of coverage would be on the y-axis here. So for example, in this region of the genome, it's covered by 1, 2, 3, 4, 5 reads. So that would be an area of great depth, but the one in the middle is only covered by 1 read. And so that's not a very deep, read. On the other hand, coverage refers to, actually this read is how much of the genome is covered by the read. So there are parts of the genome that in this case not very many, so it's well covered. So there are reads that are represented by different reads. But for example, if this reads on the right were absent, then it's not a very good coverage because the first half of the genome, you only have sequence for the first half of the genome. So those are two different categories of reading the what makes a good sequencing data. Okay, so we are about halfway. So we've covered what's a virus and just sequencing technologies. Well, these are kind of two very different topics, and then we'll put them back together. We'll connect them together with evolution and phylogenetics, which is in the second half of this talk. But before we go through that, let's take a 10-minute break. So you can grab some water, go to the bathroom, or just stretch. But whatever you do, go stand up and move. We'll be back in 10 minutes. So that would be 10:40. 10:40. Please be back by 10:40 or 10:38. How about that? All right, we'll be back.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat
Anumang hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa ay susulong. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat, lilipad. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparana sa bayang Pilipinas. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Tumitilaok na ang manok, hudyat na ng
Okay. We'll start in a few seconds. Let me just start sharing my screen. Okay. All right. Okay, so hopefully you are back and refreshed and ready to move on to the second part of this lecture. So which is on evolution and phylogenetics. And then we'll put everything together, looking at SARS-CoV-2. Okay, so first off, again, I like to start with definitions, so just so we are on the same page. So what is evolution? So evolution is the change in heritable characteristics of biological populations over successive generations. And so one of the the really influential people that started is Charles Darwin, and this is the copy of his book Origin of Species. So it's really foundational to evolutionary biology. And so evolution occurs when processes such as natural selection and genetic drift act on genetic variation. And because viruses are also biological entities, viruses evolve too. And so it's important to be able to understand how viruses change over time, which we've seen over this pandemic. It's important to also understand what is evolution, right? Okay, so back then in Darwin's time, they didn't know what viruses was, but I think if Darwin lived today, Darwin would have loved viruses, and that's because viruses are the best examples of evolution by natural selection. And specifically for RNA viruses like SARS-CoV-2, evolution can be really rapid. Their genomes change rapidly, and their life, the generation time is what's called, is really short. And so we can see in real time evolution happening, as opposed to, let's say, for example, the different finches or changes in like organisms such as mammals or birds. It takes years, decades, maybe centuries to see any appreciable change. But all of that is compressed in time with viruses. Okay, so, and that's because virus genomes mutate faster. And here's just an example, an illustration of that. So on the x-axis is the genome size. So larger gene, and then on the y-axis is the mutation rate. So you can see the trend is generally, if you have larger genome sizes, your mutation rate is smaller. So remember, viruses have smaller genomes, and especially for RNA viruses, their mutation rate is a lot higher than, let's say, bacteria, which is on the lower end, right? And that's why we can see changes much faster, and also look at virus evolution in real time. And so replicating viruses produce large numbers of mutant genomes. So what do we mean by this? So evolution is really only possible when you have mutations that occur in a population. So if the population, the virus population, is all the same, then evolution change cannot occur, basically. So it's really important to have mutations in the gene pool so that you can change the genome in time. And then these mutations are produced during the copying of any nucleic acid molecule, which may be DNA or RNA, depending on the virus. And remember, viral genomes are always mutating. There's always change. So every replication cycle, there's always some small changes in the viral genomes. And also, really important, not all mutations change in result in a change in virus characteristics. So there are actually mutations that are detrimental to the virus, and so that change, that virus lineage, will be eliminated. But there are also some mutations that don't really do anything. So they're very what's called neutral. So they can be detrimental, they can be neutral, and they can also be advantageous. So it really depends. It's not always that if you have a mutation, there's always going to be some sort of positive or a selective advantage, or what we call it in evolution. Okay, and so okay, so there is a question in the chat, which let me see if I can read. All right, so there's a question in the chat from Harold Panisa from CNU asking, what are the common reasons for mutation? Okay, so yeah, one of the most common reasons for mutation is that actually biology is not perfect, right? So every like it's not a photocopy. Oh, excuse me. Each replication cycle, whenever you replicate anything, really, and that includes if you know if and that includes our own cells, viruses as well. The mutation always happens. So in viruses, there's mutations generally for every replication cycle, as opposed to like humans. So it's part of being a biological entity. There's always error, or because error implies some sort of negative change. There's always variation, right? So it's kind of like you are genetically similar to your parents, but you're not exactly the same copy. In that sense, like there's always variation within human populations, and that's very fundamental to biology. So that allows you different biological entities to adapt to whatever circumstances that they're in. So yeah, reasons of mutation, it always happens. It's part of being a biological entity. But mutations. Okay, that's actually a really good question. Okay, thank you for that. All right, so going back to the lecture, so the public is actually constantly confronted with the reality of virus evolution. So the pandemic, the SARS-2 pandemic is one, but there's the general consensus is that a lot of emerging diseases are actually viral diseases, and these includes AIDS, the West Nile virus disease in the United States that started in the 1999, early 2000s, hepatitis C, Ebola virus disease, Zika, and COVID-19, which is caused by SARS-CoV-2. Right? And so every year we also have the evolution of our flu viruses, influenza, and common cold viruses. So that's also another product of viral evolution. And also another product of viral evolution is drug resistance to HIV. So virus evolution is faster than many can comprehend. So that's why we're talking about it now. And it can directly affect us, especially for those viruses that are of public health importance. And so like going back to the whole to the sequencing. So sequence information, if you remember, contains information of the genome. So you can think of it as a really long letter sequence that represents the genome. And this genome is heritable and therefore can inform evolution. So we'll see a lot of this in the next couple of slides, but this on the left is Darwin's first evolutionary tree. And it's apparently a really popular tattoo. So I pulled some off of Pinterest. So yeah, I personally know at least 2 people who have this tattoo. I also know a lot of biologists, so it might be by example. Okay, and so putting together evolutionary relationships it is a field that's called phylogenetics. So phylogenetics is from the Greek word phyle, meaning tribe, and genea, meaning origin. So finding the origin of this group of whatever you're looking at. And this is what Darwin was attempting in this his first evolutionary tree. And that, I think, part is part of the tattoo usually. And so the study of evolutionary history and the relationships among or within groups of organisms, including viruses, is phylogenetics. And these relationships, these phylogenetic relationships, are determined using genetic sequence, which again, like to emphasize, are heritable information. So it's passed on from the parent to the progeny. And then it's represented usually using a tree because there's branching involved. So it looks like a tree. And it's a diagram containing a hypothesis that reflects our understanding of evolutionary history. So I just want to emphasize that every phylogenetic tree is a hypothesis, meaning that it's our current understanding and can change in the light of more information or additional data. So remember, it's a hypothesis, not a it's not a static representation of our understanding of evolutionary history. Okay, so basically phylogenetic trees are kind of like family trees. And here's like actually a nice article if you ever get the chance to access. How to Read a Phylogenetic Tree by McLennan. It's really good if you ever want to learn more about phylogenetic trees. And so he or she compares, McLennan compares the phylogenetic tree as like family tree. So you can imagine like this is the great grandparents, these are their children. Some children get married and have their own kids who also get married, and so on and so forth. So you can think of this as kind of like a phylogenetic tree, really. And so the parts of the phylogenetic tree are the leaves, the branches, and the nodes. So the leaves going like piggybacking on the tree analogy, the leaves are the taxons. So in other words, each organism or group of organisms that are represented at the ends of the trees are called the leaves. And then the branches are the lines that connect, that represent the relationship between the different leaves. And then at each branch where they meet is called the node. And what the node represents is a hypothetical. We don't know what their sequence is, we know who they are. But hypothetically, it's their hypothetical ancestor at the node. So it would be where the branches meet, right? All right. And so phylogenetic trees can be represented in many ways. So you can actually flip it over. If you want to use curved lines, you can do so. You can do straight lines. You can do something like this that looks like a candelabra. You can also do that. But what's really important is not really so much of what it looks like, but what is connected to what. So you want to pay more attention to which leaves are connected by what branches. And so, for example, in this diagram, all of these trees are equivalent to each other. All of these trees are equivalent to each other. So the tiger and the jaguar are more closely related and share a common ancestor. That ancestor they share with the lion, whose common ancestor shared with the leopard, the snow leopard, and so on and so forth. And if you look at the other representation, it doesn't really matter if you flip it around or connect. The tiger and the jaguar are always the most closely related, and their ancestor is more closely related to the lion, and so on and so forth. And so, and here's just, this is not virology anymore, but I think it's useful to mention. One of, ideally, when you make a tree, all of your descendants should be should be represented by the branches. So ideally, each branch should contain all of the descendants of an ancestor. Otherwise, if you exclude some, it's called a paraphyletic group, which is not ideal. And one of the most famous ones is the old class Reptilia, which included turtles, crocodiles, snakes. But as it turns out, birds are actually part of the Reptilia. So before, when we had the old classification, Reptilia only included turtles, snakes, crocodiles, but actually birds should be included in Reptilia. Believe it or not, true story. So now Reptilia actually includes all birds. So they are the dinosaurs that live among us. fun fact of the day. Okay, so I could not make this, I could not get this to work with Mentimeter. So we'll just do this in the chat. So I have 4 trees here, and I would like for you to find which trees are equivalent to each other. So put your answers in the chat and we'll talk about that. Once people have put in some of their answers. Yeah, so you want to choose 2 trees that are equivalent to each other. So would it be 1 and 2, 2 and 3, 3 and 4, 2 and 3, 1 and 4. Okay, let's see a couple more people. Okay. Yeah, so 2 lang. Choose 3. 2 out of 4. Yeah. Okay, so I think let's see a couple more. Yeah. Okay, great. I think most people are getting it. Yeah, so the equivalent trees are trees 1 and 4. Yeah, so good job, everyone. Yeah, you are all experts. So yeah, so the equivalent trees are 1 and 4. And you can actually see there, if you flip tree 1 like around, you flip it on the horizontal, it's tree 4 essentially. So A and B are, let me close this. A and B are more closely related, and they're more distantly related to C, D, E, and F, this cluster of leaves. Great, you are all experts. Okay, so now we know how sequencing is being done. We have some idea of evolution and phylogenetics. I think what would be really interesting for all of you is using SARS-CoV-2, which is what causes COVID-19. And to understand the lineages, I think it will be useful for people here. So just to start off again, SARS-CoV-2 is a coronavirus. I think everyone knows that. And then coronaviruses have a positive single-stranded genome. And it's a class 4 class 4 in the Baltimore of viral genomes. And so coronaviruses have the largest known RNA genomes, which is about double of the next one. So it's about 30 kilobases or 30,000 letters long. The other families, groups of viruses are about half of that. So they're pretty unique in the RNA virus world. And there are 4 different groups of coronaviruses: the alpha, beta, and gamma, and alpha, beta, gamma, and delta, which I think is not shown here, but that's okay. And SARS-CoV-2 is in the beta coronaviruses in the 2B group, right? Shown here on the right-hand side. Okay, and so we'll do a little bit more Q&A. Just put it in the chat. So what is a host for SARS-CoV-2? Or I should actually start. What disease does SARS-CoV-2 cause? You can put your answers in the chat. What disease is SARS-CoV-2 cause? Yeah, that's right. So COVID-19, and then mode of transmission. So I like to ask these questions for every virus. I think it's very important. What is the mode of transmission of SARS-CoV-2? Airborne, that's right. You can also have contact, direct contact also work. Droplets, yep, that's right. And then host species. This is one interesting one. I actually should have put that last. What is the host species of SARS-CoV-2? Human, yeah, human, animals. Can you be more specific with the animals? Bats, quite possibly bats. Other animals? Mammals, yeah, a lot of mammals. Bats are mammals. Yeah. So any other animal that you've heard aside from humans? Yeah, pangolins. Pangolins actually, I don't think it's actually been shown. Yeah, shown that SARS-CoV-2 infects pangolins. I don't think I've seen data for that. But yeah, minks. Minks is a big one. Civets. I'm not exactly sure with civets, but yeah, white-tailed deer in North America. I think there was even a study that was 75% of the white-tailed deer in a certain region was positive for SARS-CoV-2 RNA. That's very interesting. Yeah, minks. And also a hamster. So there was there was an outbreak in Hong Kong that was from that originated from hamsters from a pet owner who sold hamsters. Yeah, it was the hamsters, which is, by the way, used as like a research model for SARS-CoV-2. Okay, great. You guys are all experts. So yeah. Okay, so going back, so understanding, so putting all together what we've learned about sequencing, evolution, and phylogenetics. Let's dive into the SARS-2 nomenclature. So actually the nomenclature, the different variants and lineages, they're actually based on the phylogenetic tree. So yeah, so all of this actually has all of this examination of genomes actually has implications for public health, at least for SARS-CoV-2. And so the phylogenetic tree is constructed from the full genome sequences of SARS-CoV-2. Sometimes they use spike, but usually for the lineages, they use the full genome. And so these lineages, these groups of SARS-CoV-2 can also be based on key mutations. So remember, viruses always mutate, but in some cases, mutations can occur in the same places, even though they're not related. And that's actually a strong indication that that mutation has some sort of advantage if it appears more than once or twice or 3 times. And so there are many different naming systems, but I think most people are now using the WHO, which uses the Greek alphabet. For example, alpha, beta, gamma, delta, and omicron. And here's just a table on your right, of the different SARS-CoV-2 lineages. The WHO, like I said, uses the Greek. The Pango lineage uses, which actually I kind of like, but I guess it's not that popular. That's okay. But it uses numbers. So like a B.1.1.7 would be kind of related to a B.1.1.6. So they use the branch. So each branch will have a number. And so if you have branching of a phylogenetic tree, then each branch will be numbered 1, 2, 3, 4, 5, 6, and so on and so forth. So this would actually, I think it's kind of descriptive, but it's not very catchy though. the Pango lineage. Okay, and then there's also other systems. The GISAID actually contains a lot of the full genome sequence of SARS-CoV-2. If you want to look at the sequences, you can register for an account. It's totally free. But they also have their own naming system, and also NextStrain, which is a website that's managed by Trevor Bedford from Fred Hutch. And you can also look at the sequences there. They also have their naming nomenclature as well. Okay, so all right, so some key terms for understanding lineages. Again, a mutation refers to a change in virus genome. And then specifically for for the lineages, mutation, when we say it's a mutation, it's often a single change in the letter of the sequence. But there are changes, for example, in Omicron and I believe Delta, that's a deletion of like a couple of, a deletion of about, I would say like 12 nucleotides, but don't quote me on that. Mutations can often happen in viral genomes, but like I would like to emphasize again, not all mutations can cause changes in virus characteristics, but we can use them to determine relationships between different groups. And so a lineage is a group of closely related viruses with a common ancestor. So you can think of it like a branch that has several leaves. And then are those that viral genomes that contain one or more mutations that are common in that variant. And then CDC, so the Centers for Disease Control in the United States and the World Health Organization, variants can further be classified based upon the known or predicted changes in the virus due to in the virus genome. And here they are. So here's there's a lot of words on this slide, but I figured I will give you like a definition for that. So you can think of the different levels as different levels of certainty that these mutations actually have a known effect on virus characteristics. So at the first year are variants under monitoring, which are variants that have changes that are suspected to affect virus characteristics. So that may indicate that it perhaps in the future it can be a risk. And then on the second level is the variants of interest, which have changes that are predicted or known to affect virus characteristics. So here, compared to the variants under monitoring, we actually have maybe some data in the lab that show that perhaps this change increases binding to the virus receptor or maybe escapes recognition by certain antibodies. But at the highest level would be the variants of concern, or the VOCs. And these are the variants that have been demonstrated that is associated with either increased transmissibility, or a detrimental change in the epidemiology of COVID-19, increase in virulence or clinical disease, or a decrease in effectiveness of public health and social measures, including diagnostics, vaccines, and therapeutics. So it can be, for example, the Omicron has a deletion in the spike that prevents recognition by certain types of diagnostic tests. So that's why it's a variant. It's a variant of concern. There's also some changes that are related to escape with antibodies that are used as therapeutics for COVID-19. And so in the world, there's only 5 so far that are variants of concern, and these are the alpha, beta, gamma, and delta and Omicron so far. And like I said, all of these characters, or this classification of variants, is really dependent on our ability to sequence viruses and to share them with the world so everybody knows what's going on. So that's why it's super important to have surveillance and sequencing of SARS-CoV-2, especially in a pandemic setting. Okay, so what does this look like actually? So here's this is from NextStrain. I like NextStrain because I think they make really nice figures. This is as of Tuesday, and showing you the different relationships of the different variants of concern. So you can see here that it's like the naming of the Greek alphabet is by time. So it's not sequential is what I'm trying to say. But for example, like the eta and the delta variants are resistant to the other lineages of SARS-CoV-2. And then one thing that you also notice is that if you have long branches, so kind of like in Omicron, like this, if you have long branches, it means that there's actually a lot more changes that are seen between the other lineages. So for example, Omicron has a lot more changes. Actually, that's very interesting about Omicron. And you can reflect it. It's reflected here as long branches. And you can also hear, for example, as well with the Delta variant. So the Delta started to be detected around the last quarter of 2020. As the epidemic or the Delta wave, the Delta surge emerged, there's more and more changes. So the branches are getting longer, and longer branch. And in this case, the length of the for this tree, the length of the branches actually means something. The length, it becomes longer and longer. So there are more and more changes, even in the Delta lineage. So yeah. So you can go to nextstrain.org and play around with it. You can look at different lineages and stuff. So feel free to do that on your spare time. It's very entertaining if you like that sort of things. Okay, and then here's also just a nice graph that they have. Just looking at the turnover of variants over here, saying March 2021. There was an alpha wave that was taken over by the Delta, and then eventually now Omicron. So you can see that there's turnover of the variants over time. Although we don't really fully understand why this happens, this is actually not very surprising, and it's very common with pandemics and outbreaks that you have turnover of different variants. So here's just a nice illustration. Okay, so just to wrap everything up, I hope I've imparted with you some of these take-home messages. So the viruses have nucleic acid genomes that can be DNA or RNA and are represented by a sequence of letters represent the 4 different bases. Also, the specifically rapidly change relative to other organisms through mutations in other organisms, and that we can determine, because the changes in the genome are heritable, we can determine evolutionary relationship between different viruses and different groups of viruses. And this this study is what's called phylogenetics. And that virus evolution and phylogenetics, which are interesting in itself, I think it's super interesting, but it can also be applied to public health, especially now that there's an outbreak. It can inform, let's say that there's a new variant coming, and we can expect that it is possible that this variant can emerge. But like I said, not all of the changes that we see are can translate into changes in the virus characteristics. And that's why it's important to continue to have sequencing efforts during a pandemic and continue doing surveillance. And that is all I have for you. Here's the resources, the different websites. Like I said, Vincent Racaniello's website is great. This Week in Virology podcast is great. It is 2 hours long, but you can do it while doing dishes or doing laundry. Viral Zone is also a good resource to get basic information about different viruses. iBiology, their next generation sequencing module is fabulous if you want to learn more about next generation sequencing and the details of how it's done. And then for the SARS-CoV-2 variants, we have the next strain, the GISAID, if you want to be more specific, but WHO and CDC have great resources. And my email is below. Feel free whenever if you have any questions. And then last but not least, I will stop sharing and go back to Mentimeter and ask you one final question. Excuse me. Okay, so hopefully, all right, so what is the one interesting thing that you learned today? And you can just type in your answer because I would be interested in what you thought. So we'll give people a couple of seconds to answer that question. To answer Menti, go to menti.com and then use the code that's on the top of the bar. Yeah, so there's someone who put in the chat, viruses are constantly changing. Very true. Yeah, there's viral evolution. What other things did you find interesting? Any nuggets of insight you have? Okay. Genetics. No matter how small a molecule is, like a virus, it can change the course of human history. Yeah, that's interesting. Yeah, viruses are cool in that sense. Okay. Okay, what other COVID variants and trend on how certain strain dominates in terms of infection at some point? Yeah, birds are phylogenetically closer to reptiles. Yes, that is my public service announcement for today. That birds are reptiles. Yeah, prediction variants. Yeah. Virus replication evolution, an idea for a tattoo. Yeah, I actually know a lot of people who have the Darwin tattoo. Great. These are all great. Great insights. Okay, we'll keep that open. And while that is going on, I'll keep that screen open. If you have any questions, you can type them on the chat. Or I think the moderator, I shouldn't take the moderator. The moderator can also take your questions. Yeah. Yeah, so there was a message in the chat that mentioned that tigers from the New York Zoo was infected by COVID-19. Yeah, that's true. And I think there was also other animals. I forget which ones. Okay, thanks guys. Okay, so we also have a question in the chat. Oops. About is it possible to do viral metagenomic study using Sanger sequencing machine? Yeah, so it used to be that viral metagenomics was done by Sanger. It's just, you can do it, but it's oh, that was a direct message to me. But that's okay. You can do it, but it's very the scale that you get from like the high throughput sequencing is nowhere near what you can get for Sanger sequencing. It will be so like just as a reference. So like the human genome was the first reference of the human genome was through Sanger sequencing, and it took them years to finish the draft. But now you can do it in a week possibly because of the whole like parallel. You can do sequencing a lot of like millions, billions of reads in parallel. That's why you do high throughput sequencing. There's also a question is hybrid assembly, and this is from Mike, is hybrid assembly doable with viral genomes? I'm curious because bacterial genomes do a combination of short and long read sequencing. Yeah, so you can do hybrid assembly if you have like a really, especially for DNA genomes that tend to be bigger and also have some tricky regions, especially the repeats. It's really difficult to resolve with short read. You can also do that with long read. You can do a combination. And then one thing that's also that you can do with long read sequencing is you can actually link which mutations that are selected together, which is if they're far apart or more than 100 or 200 bases apart, it's very difficult to be certain that those two mutations are linked together using short read sequencing. But yeah, so that's also one use of the long read sequencing. Okay, so for the phylogenetic method, Alan also asks, what is the phylogenetic method used in the phylogenetic tree for SARS-CoV-2 presented earlier? I am not actually sure with next strain, but I would go to the next strain. I'm not positive like what they use in the next strain, but it's called what's called a time scale. So if you look at the x-axis, they also take into account when the virus genome was the sample was obtained. So you can do that. But whether or not is like the maximum likelihood of patient, I'd have to look at the methods of next strain. I don't have it at the top of my head. I'm sorry. Yeah. Okay. So another person asks, is virus sequencing is very important to monitor today's status of COVID-19 and virus genomes rapidly change. Oh, okay. This is probably in response to what's the most interesting thing that you learned today. Okay. So another person asks, so far, considering that there are other SARS-CoV-2 variants, but not of concern yet, do we predict these possible mutations or characteristics? Do we prediction of mutation for these and possible physiological implications? Yeah, like with the help of database tools. Yeah, so this is a really good question, and this is from Lawrence. Yeah, so actually I attended a seminar earlier today exactly about this, of how there are statistical methods to determine whether or not a mutation, whether or not what's called a mutation is positively selected. So if a mutation has advantage, then it's what's called positively selected for. Then there are statistical methods to use it. So remember, not all mutations can be detrimental, neutral, or advantageous. And there are statistical methods that can distinguish those that are neutral, those that are detrimental, and those that are advantageous. And yes, it is being done. And in this case, there are also, we know a lot more about SARS-CoV-2 now than before. So especially for changes in the spike, we know which regions affect receptor binding or affect antibody binding. So if there are changes, then we can generate hypotheses and test those hypotheses in the lab, using binding experiments or neutralization experiments using different things. So yes, definitely something people do, and it's essentially what virologists do, SARS-CoV-2 virologists do today. Okay, another question from Divina. What factors affect or trigger the change of virus characteristics after mutations? Okay, So yeah, so this is actually a pretty important question. It's like, how do mutations translate to changes in virus characteristics? And that could really be that's basically what the main goal of molecular virology, right? So looking at what changes in the viral genome result in changes in virus characteristics. And yeah, it's essentially like if a mutation has some advantage, if it has some advantage, it's possible that it doesn't, then that is or I should say, if a mutation is in a place where it affects the function usually of a protein, then for example, you have we know what's called antibodies. So antibodies are molecules that are made by our immune system that can eliminate the virus. We know that there are certain regions that the antibodies for spike bind to, right? And so, okay, I'm not sure what happened. Okay, bind to, and then so if there are certain mutations there, then we can predict to a certain extent that it may change binding of the antibody. So that's just one example. There are many other ways that viruses can surprise us a lot. So that's just one example. So there can be a million different ways that a mutation can change the virus characters. Just one example. Another example would be, for example, a change that would make a virus replicate more, for example, or can replicate in different host species, for example. Yeah, hopefully that answers your question. But that's a big question. The question that that molecular virology all wants to answer. Okay. Next question. How do you optimize sequencing if there is no reference genome available since it's a new isolate? Yeah. So this question is a good one. Yeah. So there's two main ways that you can do sequence assembly. One is called what you mentioned, reference genome assembly. So you know that this virus, for example, is in your sequence, and then you match your reads, your data to the reference. And then there's also a method called de novo. It's called de novo assembly, which is, it has no guide. It's more computationally intensive because you can see like it's like putting a jigsaw puzzle together without the picture, without the final picture. So de novo sequence, there are computational methods to do that. Yes, de novo sequencing. Yeah, but like I said, you probably need more computational resources, but it can be done. And also it's a lot easier to do de novo sequencing using the long reads because there are less pieces to put together. Yeah. Okay. So does anybody else have any questions? If not, then I can probably turn over it to our host for today. Thank you so much for your questions. Thank you for your attention. This has been wonderful having you.
Thank you, Doc Leyson, for that wonderful and comprehensive lecture. And I'm sure our participants gain a lot from this webinar. If you guys have more questions to Doc Leyson, you can type it in, or you could email Doc Leyson directly. She gave her email address earlier. So for that, we would also like to commend our participants for their cooperation and participation during our discussion. Yep. And if you don't have any more question, I think we could present our certificate of appreciation to Doc Leyson. So, all right. So, Republic of the Philippines, Department of Science and Technology, Industrial Technology Development Institute, given the certificate of appreciation to Dr. Christina M. Leyson, Balik-Scientist for shares in Bioinformatics, as part of the establishment of Virology and Vaccine Institute of the Philippines, held on February 18, 2022, via Zoom. Given by Dr. Annabel B. Brianes.
All right. Thank you so much.
Was our webinar this afternoon. We can have a group picture taking camera so that our moderator can see you and take pictures of us. All right, one smile. Again, another one. 1, 2, 3. Smile. And last one. 1, 2, 3. Smile. All right. Again, thank you everyone for joining our learning session today. Please answer the evaluation form that will be flashed on the screen to secure a copy of your certificate. I think someone asked about that attendance earlier. So you can have a certificate if you fill up the evaluation. And again, this is Joseph Sonia from DOST ITDI. Like and follow our page, Environmental and Biotechnology Division in Facebook page for more announcement regarding webinars, future webinars. Magandang araw sa inyong lahat. Maraming salamat po.
Maraming salamat po.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
man ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay aakyat. Hawak kamay tayo'y aangat, lipad. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparana sa bayang Pilipinas.
Recording stopped.
Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap liwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na kaibigan, huwag kang magpaiwan. Gamitin ang